In 10.2, we are going to talk about light reactions and how they are involved in converting solar energy, which we said was light energy from the sun, to chemical energy, which is sugar. And they are using ATP and NADPH, two of things that are crucial components from light reactions that go into the Calvin cycle. So the first thing is we talked about chloroplasts, which are located in plants. Chloroplasts are very important organelles that are composed of chlorophyll, which are the pigments that give the plant the color they are. Chloroplasts also have thylakoids, and they are involved in the process of photosynthesis. Without chloroplasts, we can't have photosynthesis. So the thylakoids that are inside of chloroplasts, they are the ones that are going to transform the light energy into chemical energy, and they are the ones that allow ATP and NADPH to go into the Calvin cycle in order for photosynthesis to occur. So, one thing we need to know is that light is something called electromagnetic energy, or also known as electromagnetic radiation. Basically, it means that you have electromagnetic waves due to disturbances in both electric and magnetic fields that creates this radiation. The electromagnetic energy is going to travel in waves. Now, if you look at a wave, you'll probably know that they look like this. The top of the wave here is known as the crest, and the bottom of the wave here is known as the trough. So the wavelength is going to be from one crest to another. That is the wavelength, and the wavelength is usually shown like that. And depending on the size of the wavelength or what it looks like, it's going to determine the type of electromagnetic energy that you will receive. There's something called the electromagnetic spectrum, which is basically the entire range of electromagnetic energy or radiation that we have. So this can be from the smallest, which is a nanometer, which is usually your gamma rays, to it can be the length of a kilometer, which is radio waves, which is which is the largest wavelength that you can have. There's also visible light, which falls between 380 nanometers to 750 nanometers, and this is going to be the colors that we actually see when we look at objects. And the visible light also includes wavelengths that are crucial in driving photosynthesis. So we do need visible light in order for photosynthesis to occur. You might have heard of the word photons before. Basically, photons are light particles. They are very small. They are not tangible, but they do carry energy. And the shorter the wavelength, the more energy the photon is going to carry. And if you actually think about the sun actually radiates the full spectrum of electromagnetic energy, from gamma rays to radio waves. But because of our atmosphere, we are only able to see visible light because that's all that passes through our atmosphere, which is pretty interesting. So here's an image that you can see. You have gamma rays, which are very, very small, that go all the way up to radio waves, which are very big. Remember, the sun actually radiates from gamma rays to radio waves, but the only ones that we actually see that our atmosphere allows is visible light. Visible light is needed for for photosynthesis. You can see that in visible light, the shorter wavelength is the purple or violet, and the longer wavelength is the red. But the shorter the wavelength, the higher the energy you have. The longer the wavelength, the lower energy you have. Okay, so photosynthetic pigments. Pigments, like we said, are substances that really show what color something is. So chlorophyll carries the pigment green, therefore making the plant green. But pigments are actually specific substances that absorb visible light. So this is really important. So when you have different color pigments, such as if you see a leaf that's more purple, it has a pigment that's that color, right? But how does that work? So pigments actually absorb different wavelengths. The wavelengths that are not absorbed get reflected, and that is what we actually see. So we don't see the pigments that are actually being absorbed by the material or like the plant, but we actually see what is being reflected. So if a leaf is green, it's because the chlorophyll is reflecting that green light and absorbing the other lights. If you put white light on a pigment, the color that you see is what is being most reflected. So if you have the color black being reflected, it's because that pigment is actually absorbing all the different wavelengths of light. And then, like I said, we see green because chlorophyll absorbs violet, blue, and red light while transmitting and reflecting green light. So I'll say that again. So chlorophyll is going to absorb violet. Ooh, excuse my awful handwriting. Violet, blue, and red light, and then it will reflect the green light, which is what we see. So here you kind of see a chlorophyll. You see the granum. Each individual stack of the granum is a thylakoid. Light comes in, and the chlorophyll that's inside of the granum or inside the thylakoid absorbs specific light and reflects the green light, and the reflected light, which is green, is what we see. Now, there are 3 types of pigments in chloroplasts. There's chlorophyll A, chlorophyll B, and then something called carotenoids. Chlorophyll A tells us that violet, blue, and red light work best for photosynthesis since they get absorbed, and green is what we actually see. So chlorophyll A is the key light capturing pigment. Chlorophyll B, on the other hand, is just an accessory pigment, and then carotenoids are a separate group of accessory pigments. So since we talked about electromagnetic spectrum, there's another spectrum called the absorption spectrum. Let me use a different color. So the absorption spectrum is basically a graph plotting a pigment's light absorption versus the wavelength. So an absorption spectrum of chlorophyll A tells you that violet, blue, and red light actually work best for photosynthesis, which I just told you in the last one. And then there's also something called an action spectrum, which literally plots the rate of photosynthesis versus the wavelength of different waves. So these 2 spectrums, along with electromagnetic spectrums, play different roles in comparing the light being absorbed with different wavelengths and how that can influence photosynthesis. Now, accessory pigments that we just talked about, called the carotenoids, they can actually be different shades of yellow and orange because they absorb violet and blue-green light, okay? So I'll write that here. So they absorb violet and blue-green light. And they can also broaden the spectrum of colors that help to drive photosynthesis. Some carotenoids also function in photoprotection, which means that they just take in excess light that would actually damage the chlorophyll or react with oxygen, and can be dangerous for the cell. Because if they reacted with oxygen, they could become really reactive and cause the cell damage. Carotenoids are important for the human eye. That's why they say eat a lot of carrots, which have a lot of carotenoids, which will help in the function of your eye to make your vision well. Okay. There's something called excitation of chlorophyll by light. This is when you have a pigment that takes in light from the sun and it goes from a ground state to an excited state, which can be pretty unstable. Now, when the excited electrons that have gone from the ground state to the excited state fall back to the ground state, the energy that they have, the excess energy I should say they have, is actually released as heat. And when this happens in isolation, some of the pigments that go through this process will actually emit light and cause a fluorescence kind of glow that occurs later on. So here is an example of what I'm just talking about. So you have a chlorophyll molecule, which carries a pigment. Let me write that a little bit better. So you have a chlorophyll molecule that carries a pigment. You have a photon, which carries light energy. It hits the pigment, which excites an electron, shooting it from the ground state all the way to the excited state. As the electron falls back down, it's going to release some heat, and it can also release some fluorescence. And this is what you will see in fluorescence, almost like a glow. There's something called a photosystem, which is basically a reaction center complex that you associate with light harvesting complexes. So let's talk about this. A photosystem is composed of a reaction center complex that is surrounded by a light harvesting complex. I have a diagram of this so that I will show you. The reaction center complex is basically made up of proteins that keep a pair of chlorophyll A molecules and a primary electron acceptor together. So the reaction center complex is made up of a pair of chlorophyll A molecules and a primary electron acceptor. So if we look at this diagram, what you are seeing right here is pretty much a photosystem, right? So you're seeing a light harvesting complexes, which right here, and then you're seeing a reaction center complex also in the middle. So the photosystem is made up of light harvesting complex and a reaction center complex. And within these complexes, we're going to see what exactly happens when you are forming, when you're creating, an excited electron, or you are having light reactions occurring. When we talk about the reaction center complex, remember there is a primary electron acceptor as well as chlorophyll A molecule. Remember we said that there's two chlorophyll A molecules as well as a primary electron acceptor that makes up a reaction center complex. So just make sure you know that. I'll write that on this side. So a reaction, RXN is reaction center complex, is made up of a primary electron acceptor, which is this purple thing here, plus it's made up of two chlorophyll A molecules. And then the reaction center complex as well as the light harvesting complex both make up a photosystem. Okay, so a light harvesting complex is made up of a pigment molecules that are attached to proteins, okay? So light harvesting complex is made up of pigment molecules that are attached to proteins. The light harvesting complexes will also transfer the energy that they get from photons to the chlorophyll A in the reaction center complex. So I'm going to show you that in a second. Then the chlorophyll A molecules, because they can transfer energy to an excited electron, which can also then be transferred to a different molecule. And when they transfer that electron that now has energy to a different molecule, we'll see a reaction taking place. So again, we see this diagram. We see that the photon is coming in. And when it comes in, it transfers energy to the chlorophyll A, which is right here. There's two of them. The chlorophyll A will then cause an electron to be excited, and it will reach the reaction center complex, which is this purple box right here. And that box will now have the energy from that electron. The primary electron acceptor that is part of this reaction, it will take those excited electrons and it's going to be reduced, okay? So remember, reduced means it's going to gain electrons. So when you have this transfer of an electron from a chlorophyll A molecule to the primary electron acceptor, this is considered the first step of the light reaction, okay? So solar power transfer of electron from chlorophyll A to the primary electron acceptor is going to be the first step in the light reaction. Now, there are two types of photosystems in the thylakoid membrane. Okay, so there's two types of photosystems in the thylakoid membrane. Both photosystems are part of the light reaction of photosynthesis. All right, so both of these types of photosystems that we see, that we're going to talk about, are part of the light reaction that we're going to talk about of photosynthesis. So the first one is called photosystem 2. Why it's called photosystem 2 first? You can ask the scientists who came up with this. So the photosystem 2 actually functions first. The reason that they say that they call them photosystem 2 or whatever is because they reflect when they were discovered. But moving on from that, photosystem 2, like I said, will function first in a light reaction. And this is going to have the reaction center, chlorophyll A, so the reaction center of chlorophyll A in photosystem 2 is going to be called P680. And this is going to be best at absorbing the wavelength of 680 nanometers. That's why it's called P680. So when you have a light reaction, the first photosystem that's going to act on it is photosystem 2. And this is going to be also called AKA P680 because that is the wavelength that it absorbs. Photosystem 1 is the best at absorbing P700 nanometers, and this will happen after photosystem 2. And so it is going to be called P700. The reaction center of chlorophyll A in photosystem 1 is going to be called P700. So photosystem 1, aka P700. And then remember photosystem 2, which will happen before photosystem 1, is going to be called P680, reflecting the wavelengths that they absorb. Okay. When we talk about linear electron flow, this is what happens during a light reaction when there are two possible pathways for an electron flow to occur. They can either occur cyclic or linear. So remember, electrons are the ones that are being energized to go from a ground state to an excited state to kind of give energy to the reaction center. So there are two ways they can happen, cyclic and linear. Linear electron flow is when you have a flow of electrons through the photosystems and other molecular parts in the thylakoid membrane, okay? So linear flow is also going to be the primary pathway. It's going to involve both photosystems, and it's going to make ATP and NADPH using light energy. Make sure you know this, okay? Again, linear electron flow is the primary pathway. It's going to use photosystem 2 and photosystem 1, and it's going to produce ATP as well as NADPH using light energy, okay? So that's an important thing for you to know. There are 8 steps that are part of the linear electron flow, all right? So we're gonna go through them. The first one is that you have a photon that's going to hit a pigment in the light harvesting complex that we saw earlier. You can follow along in your textbook. They have really good diagrams. So you're going to have a photon that's going to hit a pigment in the light harvesting complex of photosystem 2. Then the energy that is gained from that is going to be passed through the pigment molecules until it excites photosystem 2, also known as P680. The electron that is going to now be excited from P680 is going to be transferred to the primary electron acceptor that we will now call P680 plus. Okay? So that's what you see here. You have light coming in through a photon. It's going to hit these pigment molecules that are going to pretty much pass the energy until it reaches a P680. That P680 will excite an electron until that electron will reach a primary electron acceptor. This primary electron acceptor is also going to be known as P680 plus. And then it will be reduced by an electron. The third step is you are going to have an water molecule that's going to be split by enzymes, and the electrons that are going to be coming and transferred from the hydrogen atoms of the water are going to be moved to the P680 plus. Therefore, that is how P680 gets reduced, okay? So electrons from the water molecules split the photons and excite the electrons, therefore reducing P680 plus to P680. P680 plus is also the strongest known biological oxidizing agent. Remember, oxidizing agent means it gets reduced, which means that they gain electrons. The hydrogen ions are going to be released into the thylakoid space within the chloroplast, and then the oxygen is going to be released as a byproduct in this reaction. So that's what we see here. Okay, so while you're having, in step 1, light's coming in, transferring energy through the pigment molecules then cause energy transfer to P680, which excites an electron to the primary electron acceptor. You have water that's going to come in that brings the energy through the electrons, and it's going to reduce P680 plus to P680, and it's going to make 2 hydrogen ions as well as oxygen. Number 4, electrons are going to go down the electron transport chain from photosystem 2 to photosystem 1. Electrons will, from the fall, going down the electron transport chain, will cause a proton gradient, which will go across the thylakoid membrane. Okay? Then in step 5, you're going to have potential energy that was stored in the proton gradient, which is going to cause ATP production to be processed through chemiosmosis. Okay? So the potential energy that was stored in the proton gradient is going to cause ATP production through the process of chemiosmosis. And then that's what you see in this figure. You have the electron transport chain. You have chemiosmosis that's occurring. in step 5. And that's going to cause ATP production. In photosystem 1, just like in photosystem 2, you're going to have, so in photosystem 1, it has light excited electrons that's on a P700, right? So the electrons actually came from photosystem 2. So the electrons from photosystem 2 get transferred to photosystem 1, because remember, photosystem 2 occurs first. And it's going to excite the electrons in P700, which is photosystem 1. The excited electrons are then going to go to P700 plus, which is a primary electron acceptor, which then gets reduced to P700. All right, so I'll say it again. In step 6, photosystem 1 has a light excited electron that's going to be located on P700. The electrons that are in photosystem 1 are received from photosystem 2. These excited electrons are going to go to photosystem 1 and make it for P700 plus, which is going to be the primary electron acceptor, which would then become reduced and become P700. So you'll see this. You have P700 here. You have electrons that are going to be excited to become, that are going to go to the primary electron acceptor, and then it will become reduced from P700. From P700 plus, it will become P700 after it gets reduced. In step 7, after the primary electron acceptor has the electrons fall back to a protein, this will occur through another electron transport chain. So another electron transport chain is brought in, which will cause the primary electron acceptor to have electrons fall through it through a protein. Then you're going to have a protein coenzyme called NADP+, called NADP plus reductase, which is going to catalyze the electrons from the electron transport chain to make NADP plus to reduce it to NADPH. So this is going to take the hydrogen from the stroma, which is like that cytoplasm material in the chloroplast, and be used in the Calvin cycle. Okay, so I'll say that again. In step 7, your electron is going to go down the electron transport chain from the primary electron acceptor in photosystem 1 to the protein called FD, or ferredoxin. The NADP plus in step 8 is going to catalyze the movement and transfer of electrons from NADP plus by reducing it to NADPH. Then the electrons that are now being shuttled in NADPH can be used in the Calvin cycle, which, remember, we said that the light reaction supplies NADPH as well as ATP for the Calvin cycle for photosynthesis to occur. And this occurs because hydrogen is removed from the stroma. And that's what we see right in this diagram. Electron transport chain continuing to happen. You see the protein right here. You see the electrons moving down. You see NADP plus being reduced to NADPH to carry electrons into the Calvin cycle. So I know this is a lot. The best way to do this and study this is to go through your book, review the diagrams and review the steps that go with it, and really understand it, especially in your outline. Make sure you have all these steps in the outline. Sometimes it might help to actually draw the diagrams as well. Okay, so as electrons go through the linear flow, they have energy changes that can be seen as a mechanical kind of analog, right? So you can see that the photon brings in the light energy, excites an electron. The electron will go down the electron transport chain, make ATP along the way. Then you have more photons that come in, excite another electron, then will cause the movement of the electron going down another electron transport chain to make NADPH that will then go into the Calvin cycle. So So you have two electron transport chains that are really happening. And remember, it always goes from photosystem 2 to photosystem 1. So now we just talked about linear flow, which is the primary passageway. There's also something called cyclic electron flow. This is when the electrons cycle back from FD, which is a protein, to photosystem 1 through something called the plastocyanin molecule, also known as PC. So instead, remember in linear, we went from PS2 to PS1. In cyclic, you're going to go the opposite pretty much. And so in cyclic, electrons flow uses only photosystem 1 mainly. So photosystem 2 isn't really going to be a part of it. But it's going to make ATP, which is also needed for the Calvin cycle, and not NADPH. So remember, linear cycle makes NADPH, cyclic electron flow or cyclic cycle is going to make ATP. And then you're not going to have any oxygen being released in this process. So again, cyclic electron flow, you are going to pretty much only be using photosystem 1. When I say it's the opposite of linear, it's because you're not going this way. It will kind of just circle back and forth between PS1 and itself. And it's going to make ATP instead of NADPH because NADPH was made in the linear flow. So you can see how in cyclic electron flow, you have photosystem 1. And instead of a linear flow where you have photosystem 2 going to photosystem 1, it kind of just revolves within itself. So you have the primary electron acceptor, you have the protein FD, you have a cytochrome complex, which is going to release ATP, and then you have a PC, which is a molecule that's going to help excite the pigments in order for electrons to be fired up and excited. So in Calvin electron flow, you have electrons that are going to cycle back from FD to the cytochrome complex to PC to P700 and then to the primary acceptor and then back again. Okay, so chloroplasts and mitochondria both use something called chemiosmosis to make ATP. And that is, one of the main ways that they function in order to create energy, even though they could use different sources of energy in order for this to happen. Mitochondria transfer chemical energy from the food that we eat. So they take the sugar, break it down from the food that we eat in order to make ATP. Chloroplasts, they take light energy from the sun and they transfer it into chemical energy, also known as sugar, and then that's how they also make ATP. Now, because chemiosmosis and chloroplasts and mitochondria differ, this also shows you the similarities between them and how they're both trying to make energy for, either a plant or for a human or an animal. So there are, differences, but also a lot of similarities between them. One of the differences being that in chloroplasts, electrons drop down from a transport chain, through the process of water being involved with it. In mitochondria, they are taken from organic molecules in order to make the energy that they need. So this is kind of just a diagram that shows you what is happening when we talk about mitochondria and chloroplasts. In the mitochondria, you have the intermembrane space, you have the intermembrane space. In the chloroplast, you have the thylakoid space and the thylakoid membrane. You can see the electron transport chain, and that's how they move hydrogen ions around. And then you have also something called an ATP synthase, which helps in the production of ATP. So they have a lot of different spaces, but you know, the main thing is they're both involved in making energy for whatever organism they're part of. So in the mitochondria, you have something that is called protons that that we've studied before, and they're going to be pumped into the intermembrane space in order to make ATP synthesis occur. ATP synthesis is just a production of ATP. And what happens is, as they're pumping these protons through these intermembrane space, they're actually allowing ATP synthesis to occur even as they diffuse back into the mitochondrial matrix. So it's kind of like a cycle that's occurring. In chloroplasts, protons are being pumped into the thylakoid space and then back into the stroma in order for ATP synthesis to occur. So we kind of saw this in the image. So I'll draw it up here. So if you have a mitochondria, you have the intermembrane space here. So you have protons that are going to be pumped from the intermembrane space back into, so let me actually pull up the diagram really quick. It might help you guys understand a little bit more. So here, we have a mitochondria. You have the intermembrane space, like I said, and the matrix of the mitochondria. So what's happening is that the mitochondria is going to have protons that are going to be pumped into the so here's your proton. It's being pumped into the intermembrane space. And as a proton is pumped into the intermembrane space, it's going to come back and make ATP. For chloroplasts, you're pretty much having the same thing as well, but it's going to happen in the thylakoid and the stroma. In chloroplasts, you have protons that are being pumped from the thylakoid space, which is over here. I'll use a different color here, into the stroma. And that's how you're going to drive the synthesis of ATP. So there's two different types of methods that's occurring, but whether it's the mitochondria or the chloroplast. So remember the two differences between a mitochondria and chloroplast, and which one has intermembrane space and which one has the stroma and the thylakoid space. Because those are going to be important when you're defining how each of them drive ATP synthesis. ATP and NADPH are going to be both produced where the stroma is, and that's where the Calvin cycle is also going to be taking place. So light reactions, if we were to summarize what we just talked about, light reactions are going to make ATP, and they're going to increase the energy in electrons by moving them from H2O, which and transferring those electrons to NADPH, which is your electron shuttle. And they will take that electron and its energy into the Calvin cycle. So remember, ATP and NADPH are both produced on the Calvin cycle side. The light reaction produces ATP and the potential energy of electrons by moving electrons from water to NADPH by carrying them on the hydrogen molecule that's attached to the NADPH. This is just light reaction and chemiosmosis occurring. You have water right here that gets split by photosystem 2. So photosystem 2 will split the water in the thylakoid space. And then you have PQ, which is going to, right here, which is going to transfer electrons to the cytochrome complex. And then 4 protons are going to go across the membrane from the stroma into the thylakoid space, which you see right here. And then when this happens, you're going to have hydrogen ions are going to be moved from the stroma when the NADP plus picks it up. And you're going to have diffusion of hydrogen from the thylakoid space into the stroma, which is going to allow ATP synthase to make ATP and allow ATP production to be made. And NADPH and ATP are going to shuttle the energy into the Calvin cycle, and that's how the Calvin cycle gets NADPH and ATP. So here's just a closer look of that happening. You see the production of ATP right here through ATP synthase, which is getting, the energy from the protons that are being pumped. So this is a lot of information, I know, but this is very important to know when you are working on, when we're learning about photosynthesis. You probably have never heard of photosynthesis being this complex, but it really is. I really encourage you to go through the diagrams in your textbook, make sure it's in your outline, because we will be checking for that, because this is something that you do need to know. Your quiz is made up of a lot of questions about these systems, so make sure you are thorough with it and can answer the questions correctly.